Okay, thanks, Francois, and it's a pleasure to present to the CNSP. What I'm going to talk about, formal title is Scientific Platforms in the Development of an mRNA Vaccine Delivery System, but really the an alternative title, and one that I'll use throughout, is a 25-year journey to enable gene therapies, which led in part or to some extent to the Pfizer-BioNTech COVID-19 vaccine. I have a couple of conflicts of interest. I found that they're germane to this presentation. I'm a founder of Precision Nanosystems, and I'm founder and chairman of Acuitas Therapeutics. Okay, just to get down to some basics. What is gene therapy? And according to Wikipedia, it's a very general definition. Gene therapy is the therapeutic delivery of nucleic acid into a patient's cells as a drug to treat disease. The operative word there is delivery, and so that's the platform technology really that I'll be talking about throughout, which is a delivery technology to deliver nucleic acid polymers into a patient's cells. And so this is required for very basic reasons. The unmodified nucleic acid polymers are very rapidly degraded in any biological fluid. If you inject them systemically, they don't preferentially accumulate where you want them to go. They may not get there at all. And even if they get there, they can't penetrate into target cells because they can't get across the cell membrane. Now, this is an area of really extreme interest, gene therapy, because potentially any disease can be treated using this approach. And that's resulted since about 1990 in 2,500 now, I think, gene therapy trials. Most have used viral delivery systems. However, out of all of that work, it's an enormous amount of work and enormous expense. There's only been 6 drugs approved so far. 3 of those have used viral vectors, adeno-associated virus, and so those are the 3 indicated: Chimera, Luxturna, and Zolgensma. One of those is ex vivo Chimera for CAR T-cell therapy. But now there's 3 others that have arrived on the scene quite recently, Onpattro and 2 COVID-19 vaccines. And I'll be describing the Onpattro and Pfizer BioNTech vaccine throughout this talk. So it's a situation that's changing. And why is it changing? The viral vectors, there's been a tremendous amount of really amazing work trying to get viral vectors to be more suited to gene therapy applications, but they suffer from a lot of issues. Limited genetic capacity. They're quite difficult to engineer and manufacture. They'll stimulate an immune response, which means that you can only give 1 dose. There's a variety of other issues, very expensive, toxic, can give rise to mutagenesis. And so it's been a fraught journey for viral vectors as gene therapy platform technologies. Non-viral systems potentially have none of those issues, but historically, they've had limited transfection potency and can be quite toxic in vivo. And so these issues are now being overcome using lipid nanoparticle delivery systems. And I'll go through a little bit in this talk as to how that has happened. So lipid nanoparticles that enable gene therapy. I'll describe quite briefly the design of these systems, and then go on to the Onpattro story. This is a drug to treat a condition known as hereditary amyloid transthyretin-induced amyloidosis, and just the program and the disease, and then the clinical trial results. And then I'll close with a few slides on the BNT162b2 story. This is the Pfizer-BioNTech COVID-19 vaccine. Now, going back to 25 years ago, this was the challenge that we took on, essentially to develop a delivery system, a lipid-based delivery system that takes nucleic acid-based drugs, from to the liver at so by an intravenous injection, taking these polymers to the liver, getting out of the vasculature, and then being taken up by the cells, and of course then releasing the contents inside into the cytoplasm. So this is quite a ambitious objective. And of course, your immune system is exquisitely tuned to try to make sure that this never happens in terms of getting genetic information into cells in your body. So just to start off in a very basic way, if you want to encapsulate a nucleic acid polymer, indicated by the double helix here, in a lipid system, then the simplest way to do this efficiently is to have some positively charged lipids there that will associate with a negative charge, and you can see intuitively how you'll end up with a hydrophobic entity that conceptually can be placed into a into a lipid-based system. Now, there's no cationic lipids in nature. They're extremely toxic molecules in biological systems. There only are net neutral lipids or negatively charged lipids. And the problem that these positively charged lipids have is that they'll combine with negatively charged lipids and essentially rip membranes apart. And so we were faced with the issue of how do we get an encapsulation using positively charged lipids without using lipids that are going to be so toxic as permanently positively charged cationic lipids. And so what we did was we tried a lipid called DODAP, which we had in the lab for some basic science reasons. This is an ionizable cationic lipid, and that just means that when at pH 7, or 7.4 physiological pH, it's net neutral. However, the primary amine gets protonated as you go down, say, pH 4. The pKa here is about 6.5 or so, and becomes positively charged. And so you have a system that at pH 4, about the acidity of a lemon, is positively charged, but is not charged at physiological pH, and so therefore is much less toxic. Now, what we found is we could formulate nucleic acid polymers into the lipid nanoparticles using a rapid mixing procedure, essentially where we dissolved all of the lipids, and this is just, there's 4 lipid components in addition to the ionizable lipid that's indicated in green here. There's a PEG lipid, polyethylene glycol lipid, and a structural lipid, phosphatidylcholine and cholesterol. So we mix those together with water at pH 4, buffered at pH 4, with the oligo that we want to encapsulate in that medium. Now, very rapid mixing, one of the first things to fall out of solution is going to be this hydrophobic particle here, where you have the cationic lipid, the ionizable lipid, that's positively charged, interacting with the negative charge on the oligonucleotide. If you do this very slowly, of course, they're going to aggregate and you get a great big glob of material. But if you do it fast enough, you can trap them in limit size systems. And essentially, the lipids such as the PEG lipid will fall out of solution. These are surface lipids, and really stabilize the whole system in these limit size particles. Then we dialyze away the ethanol and raise the pH. And so this resulted in a class of lipids. This is indicated here by cryo-TEM. This bar here is 100 nanometers, so they're about 60, 70 nanometers in diameter. They have a hydrophobic core. This is a cartoon form here that contains the oligonucleotide RNA, SIR, small interfering RNA or messenger RNA, whatever you might want. And so they're ideally suited to encapsulation of macromolecules, negatively charged macromolecules. We can get encapsulation efficiencies approaching 100%, stable, monodisperse, relatively non-toxic, as I mentioned, and highly reproducible. So this was the formulation side of it. Now we move on to the application for this disorder that I mentioned, transthyretin-induced amyloidosis. So this was our objective from about 2005 to 2012, is to develop these systems using siRNA to silence genes in the liver following an IV injection. Obviously, there's a lot of disorders one can target using a if you can silence or express or edit genes in the liver. So, can these systems that contain the ionizable lipids, such as this ionizable lipid DODAP, can they silence genes in hepatocytes? And so they're quite simple systems. There's no targeting information on the outside. And as we discussed, the oligonucleotide is encapsulated in the hydrophobic core. Now, we assess the in vivo potency of these systems using a factor 7 model. Factor 7 is made in hepatocytes, it's secreted into the circulation. It's one of the clotting proteins. And so what we did here was to inject lipid nanoparticles containing siRNA to silence factor 7. So this gave a relatively straightforward assay whereby we could inject the lipid nanoparticles and then 24 hours later assay for factor 7 in the blood. And so the extent to which the levels of factor 7 were knocked down would reflect the potency of the system. So this was a reasonably high throughput model. We could run 2 experiments a week and to go through a number of formulations, a large number of formulations to develop the best or the optimized systems. This just indicates the range, the types of ion. What we found to our surprise, actually, was that the potency of these systems was extraordinarily sensitive to the species of the ionizable cationic lipid that we employed. We synthesized over 300 different species in this collaboration. This collaboration was done with a company in Boston called Alnylam. And we moved through to these to MC3, which has ended up as being the most potent. But you'll note really that these are not very different molecules. I mean, they're quite simple, quite similar. And the only difference primarily is in their pKa. But this just shows you the remarkable improvement in potency that we're able to achieve over this 5-year time frame. Moving from a dose of 10 milligrams of siRNA per kilogram body weight to get 50% gene silencing. That's what the red line is here. I cut off the y-axis there. But anyway, to move from the dose level of 10 mg per kg down to 5 micrograms of siRNA. So, you know, over 1,000-fold improvement, 3 orders of magnitude improvement in potency. Without increasing toxicity. So it was we ended up with a therapeutic index of over 1,000. So in other words, you give 1,000 times higher dose before we saw toxicity. So the results of this program was that we could silence the factor 7 gene using these lipid nanoparticles containing siRNA to silence factor 7. And by extension, we can silence any gene we want in hepatocytes. And so it's just a matter of putting in a different siRNA to silence that will silence a particular target protein. So the clinicians at Alnylam chose to develop the system, lipid nanoparticle system, to treat this transthyretin-induced amyloidosis. This is a disorder that affects about 50,000 people worldwide. It might be more actually, but these are people who are actually diagnosed with the disorder. Transthyretin is a protein that's synthesized in the liver, secreted into the circulation as a retinol binding protein. But if there is mutations in the transthyretin gene, this can lead to the formation of fibrils that will circulate in the circulation and deposit presumably in all tissues. But it has really nasty ramifications in the nervous tissue and in cardiac tissue. And so there's really no effective therapy for this disorder. It's really usually fatal within 5 years of diagnosis. It's a really nasty multisystemic disorder. This is just indicating CNS problems, gastrointestinal problems, kidney problems, neuropathies, cardiotoxicity. The neuropathies include difficulty walking, etc. Actually, that's usually one of the first signs you're coming down with this disorder. And it's rather a nasty indication because, of course, you'll have seen relatives, this is a hereditary disease, and you've probably seen relatives come down with this disorder, and the progression can be horrific, as is indicated here, over the 5 years of the course of the disorder. Now, one of the things about gene therapies is that conceptually, at least, you can develop therapies that are really very targeted to the disorder. And so in this case, seeing that transthyretin is causing the disorder, well, let's silence it in the liver. And so the hypothesis here is to deliver siRNA to silence transthyretin production in the liver, reduce the levels of circulating tetramers. The organ deposition of the fibrils, of course, will be decreased. Perhaps you'll get clearance, maybe you'll get stabilization and recovery. The main point here is it's a potentially simple solution to this devastating disorder. So there's a clinical development program started in 2012. It was phase 1 was completed on healthy volunteers. And then there was an 18-month study that was concluded in August 2017 that investigated the efficacy in patients. And so this is just the results of the phase 1 study, which established the dose and interval levels of the lipid nanoparticle siRNA to silence transthyretin, the levels that should be given. And what you note here is that at levels above about 1.15 mg per kg, you're seeing this very dramatic silencing or decrease in the levels of transthyretin in the circulation, which extend over 20, 30 days. And so on the basis of this, the clinicians chose a dose of 0.3 mg per kg every 3 weeks for the subsequent clinical trials. This now went on to the phase 3 study in about 225 patients. There was a treatment arm and a control arm, placebo. They got sterile saline. And the endpoints that were looked at here were the change in the neural impairment score at that 18 months, quality of life, weakness, ability to walk, body mass, and so on. So these are standard indications for particularly for neuropathic problems. Now, the results were announced in September 2017, and the drug in this clinical trial hit the primary endpoint and all secondary endpoints. So it was quite I think this is certainly the best result of a clinical trial that I've ever seen. So in terms of the neural impairment score, it actually improved compared to the initial condition, and so you're not just stabilizing the disorder, but you're actually making people better. P-value here, p-value. I don't know why they had to bother to calculate a p-value. There's no doubt that this drug works, and similarly, that's true for quality of life. As for self-reported quality of life, muscle strength, gait speed, nutritional status, and so on. So this Onpattro, or patisiran as it was called during development, is a stabilizing and potentially curative therapy for a previous fatal disease. This drug was approved by the FDA in 2018 for treatment of transthyretin-induced amyloidosis. It's the first FDA approval of an RNAi-based gene therapy drug, so it's a pretty big deal. So it dramatically demonstrates the power of gene therapies. So now I'm just going to go on to the last part here and so give you a few slides, or show a few slides on the development of the COVID-19 vaccine. And here we're dealing with messenger RNA as opposed to siRNA. So this is a much larger molecule. So the question here is, does the lipid nanoparticle technology enable mRNA-based therapeutics? And so it's exactly the same process that we envisaged for the siRNA, intravenous injection. If we can silence genes in hepatocytes, perhaps we can express genes in hepatocytes. So we're facilitating the intracellular delivery and then the delivery to the cytoplasm after being taken up by endocytosis. Now, these are messenger RNA. They're much larger entities, as indicated here. I've tried to do this to scale, but it's not quite. If I really did it, you would be able to see the siRNA here, which is a much smaller molecule compared to the mRNA. Now, it turns out that the ethanol dilution and rapid mixing process allows very efficient encapsulation. And this also results in delivery of mRNA that distributes proteins in hepatocytes. So this is the indication here for the encapsulation. And as you'll see, they look very similar to the systems by cryo-TEM that we had for encapsulation of siRNA. So this is, again, a 100 nanometer bar. Dosing these animals intravenously and then looking at expression in the liver, you'll see here, this is for luciferase. As you can see, this is live imaging at 4 hours post-dosing, and you can see a dose-dependent expression of luciferase in these mice. Now, this was work done by Cullis, a company I co-founded in 2009. And what they've done is gone on to develop improved lipid nanoparticle systems that are now, well, it's more than 30-fold now, get higher levels of gene expression in the liver for these mRNA systems using luciferase as a reasonably high throughput system. Now, this is where serendipity comes in. We were approached in about 2014-15 by Drew Weissman, who said, well, I need a delivery system to get messenger RNA coding for viral proteins into muscle and immune cells. Drew is an immunologist. He's been working for many years with Katerine Carrico to explore the potential of mRNA therapeutics as vaccines. And they'd found modifications of mRNA that really improved gene expression, but they still needed the delivery system to enable this to be used in vivo. And so, of course, this is an introduction of mRNA that's coding for a viral protein, and which then results in, say, muscle cells in presentation of those antigens on MHC class 1, or if they get into antigen-presenting cells, MHC class 2, and giving rise, Drew hoped, to a vigorous immune response. Some of the first work was done on Zika virus, and so this was published in 2017. So here, the mRNA that was encapsulated was the pre-membrane and envelope glycoprotein of the Zika virus. And the results were quite spectacular, that total protection against Zika virus. And so injecting this mRNA intradermally in this case, and then challenging at 2 weeks or 20 weeks. And this is basically total protection against the Zika virus infection in these animals, just from one injection of the nanoparticle containing the mRNA. Now, in January of 2020, Cullis was working with a German company called CureVac and on a rabies vaccine. And so in this case, there's an mRNA coding for the glycoprotein on rabies. And this is an actual clinical results. So CureVac announced positive results at a very low dose of 1 microgram of mRNA in this phase 1 study. So high levels of neutralizing antibodies that looked extraordinarily effective. And so at that point, Cullis was also working with BioNTech, and BioNTech was working with Pfizer on a flu vaccine. And so in January of last year, all efforts switched to developing a vaccine for COVID-19, given that the pandemic was emerging. So here, the messenger RNA is coding for the SARS-CoV-2 spike glycoprotein. And all the rest of this is history, which, of course, you're probably well aware of. This was announced in November of 2020. 95%, 94%, or it is 95% effective against COVID-19 in a great variety across all cohorts, gender, race, age, etc. Well tolerated, and it's the Pfizer BioNTech is now manufacturing here. They set up to 1.3 billion doses. I think they're saying they're going to manufacture 3 billion doses this year. It was approved for emergency use in a variety of jurisdictions, including Canada. And so this is just real world evidence that this drug, this vaccine works extraordinarily well for Israel, a country of 9 million people, just showing that when to the point where these are fully vaccinated with two doses, but you can see the impact on the infection rate in Israel, and then they're now back to normal. So it's been quite a journey from developing a system for delivery of nucleic acid polymers to the liver, to the first drug that was approved for RNAi-based therapeutics, and then to the vaccines, which were obviously developed at a very high speed. Vaccine applications are really just the tip of the iceberg. We're going to see vaccines against HIV, Zika I mentioned, perhaps a universal flu vaccine, malaria, but there's many other applications where we might need a protein being expressed that can be expressed using this mRNA approach, or edited for that matter. And then of course, there's the rare diseases that are again attackable using this type of technology. Just one point is that the process here is very rapid. Once we identify the protein that we want to silence or express or edit, we can produce the sRNA or the mRNA in a time on the order of a month or two. Encapsulation is very fast, a day or so, and have a new medicine that's available in a time on the order of a few weeks. And so this dramatically shortens the time frame for development of new highly targeted therapeutics. Just acknowledgements. This is the work of literally hundreds, probably closer to 1,000 people, but I want to particularly acknowledge Mick Hope, Tom Madden. I've been working with them for 40 years now, and one of the reasons that we've been successful is that we've held a team together over that long time period. And that also goes for people like Steve Ansel, Ying Tam, Barb Wee, Paul Lin, Sean Semple. I've worked with most of them for more than 20 years. And then there's a variety of people that are involved on the microfluidics side of things, which I didn't mention: James Taylor, Ewan Ramsey, Carl Hansen. People are Abutis, James Hayes, and Ian McLaughlin. Al Nyland, great collaboration with them. Marco Ciffolini in the chemistry department. These things work in the brain as well. My own group, and Drew Weissman, who I mentioned at the University of Pennsylvania. And so with that, I'll close, and thank you for your attention.
Okay, so welcome back. Bonjour à tous. First, I would like again to thank Pieter for such an exciting talk. Next, we'll share with you a short discussion with Pieter where he underlined his vision regarding the next frontiers in this rapidly evolving research area, which is the application of LNP technology, as well as his insight into the next frontiers in basic research. He will also discuss the importance of scientific platforms to achieve research breakthrough in Canada, such as the development of mRNA vaccine delivery for COVID-19. I would like to point out that you're welcome to forward your comments and questions for Pieter. He actually said he will be pleased to get back to you personally on your questions and comments. So I will make sure that he received them. And I would like to also point out that you should stay with us as we hear from Mia Selavi immediately after the short interview. So I thank you again for your attention, and merci à tous, and I will see you in the next session. So first of all, thank you, Pieter, for your very exciting talk. And more importantly, I would like to thank you, you and your team and your collaborator for the development of such a robust and very important technology platform, not only for gene therapy, as you pointed out, but also for mRNA vaccine development. So thank you again. So first, I would like to discuss a little bit with you the what do you think are the next frontiers and research areas for your technology? I think you listed some bullet point on potential application, but I would like to hear from you is how do you prioritize? So what do you think? Which one you think you should be investigating first?
Yeah, it's that's a good question. Because there are so many things that one could go after. I mean, obviously, there's a lot of disorders. I mean, if you can have a protein you can use the liver as a bioreactor, produce antibodies that might be therapeutic utility. You can replace proteins that aren't perhaps being made properly. But for me, the main thing right now is to get past the liver.
Yeah.
We're good at transfecting hepatocytes, but I think there's a push now to say, okay, well, can we transfect bone marrow? Can we transfect the lung? Can we get to the eye or the brain? And so those are the the areas in which we're focusing now. In order to do that, one has to develop delivery systems that circulate for much longer in the body. The systems we currently have are quite well targeted to the liver, actually, but it means that within 15-20 minutes after injection, 80-90% is in the liver. Mm-hmm. And so what we're doing now is developing systems that circulate for a longer time, and so therefore we will have access to tissues such as bone marrow, which we're starting to get quite good at. And also other tissues in the body. And I think the thing after that is going to be, well, how can you turn them on and off in one location versus another? You don't want to have a global expression of something that's maybe only needed in one tissue. And so the problems are going to continue for quite a while yet, and they're really quite fascinating ones.
Yeah. So I think from a molecular virology perspective. And keeping in mind the challenges for many years for vaccine development for HIV and HCV, I think this is certainly for those two specific viruses that can be actually transmitted or be present in the same individuals at the same time. That could be very powerful to develop such a technology. What I wonder, though, have you thought of basically targeting multiple pathogens at the same time? So, for example, by combining mRNA encoded by two different viruses, so example HIV, GP120, and perhaps E1, E2 of HCV. So multiple mRNA delivered through your technology. Is that feasible, do you think?
Well, it's very feasible, yes. People haven't really done it yet, but there's nothing to say you can't do that. Now, you do it two ways. Of course, you could just make two separate formulations and then mix them together, or you could have everything there starting from scratch. But yes, it should work, and there's intrinsically no reason why it wouldn't. As I say, right, we're in one of those stages where the gates have kind of opened, and there's just a lot of things to do, and so it's certainly created enormous excitement in the field. The amount of effort going on in lipid nanoparticles now is probably 100 times more than was in place about 2 or 3 years ago.
Yeah, another I think important area is I think you alluded to this specific one is therapeutics. So I think mRNA-based delivery is great, but obviously based on the work you've done with RNAi and even mimic of microRNAs or anti-microRNAs, one would think that this would be a very powerful approach or a strategy for developing again antiviral treatment. Is so where would this antiviral treatment strategy is compared to the mRNA-based vaccine? Is in terms of thinking about priority moving forward, is that high on the list, or this is like in the middle of the list?
Yeah, the short answer is that, well, I'm not an immunologist, but one of the, I'll give you an example of the kind of things that can be done, which actually are still mRNA based. You can make mRNA that, say, codes for the heavy and light chain of an antibody. And some of the work that Cullis has done with Drew Weissman is to say, okay, well, let's make an antibody against HIV, and then challenge the mice with HIV, and again, you get total protection. You get the antibody being expressed for 7 days or longer, and no protection against HIV. Whether that's going to be good as a therapeutic or not, I don't know. But certainly as a preventative for high risk people in high risk situations, you can see that being you have utility for say subsequent pandemics or whatever. There's a large number of different ways that you can deploy this approach. That's for sure.
You're going to be very busy, it seems to me. I think that this is there's so many applications I can see that are actually extremely important to work on. The last question on this section is biomanufacturing. Is that, I mean, is how challenging it is for your technology platform. Is like, I can imagine you probably need kilograms of those liposomes. So where do you reach out? Do you get support here in Canada, or do you need to reach out to other countries to scale this up?
Yeah, this is really a multinational scale-up process. The lipid nanoparticle formulation was licensed by Cullis to BioNTech, and then in turn, of course, when they through the Pfizer. So when they're scaling this up, one of the advantages, of course, is that the dose levels. I mean, it's only 30 micrograms of mRNA. Now, the amount of lipid that's involved is about 20 times more by weight. And that's, it's cholesterol, it's the ionizable lipid, a bit of PEG lipid, and a bit of phosphatidylcholine. So most of those, some of those constituents are quite easy to make. It's the ionizable lipid and the cholesterol that tend to be the bottleneck. So that's one of the, from a straight procurement point of view. From the manufacturing point of view, that's not really a bottleneck at all. It's turned out to be it's really quite straightforward, because you're really just mixing the lipid dissolved in ethanol with the mRNA dissolved in a pH 4 buffer. And then subsequent, so there's just a mixing process, and then you dialyze away the ethanol and raise the pH to physiological pH. So that's not, the manufacturer has not proved the issue. The mRNA has not proved the issue, to my surprise, actually. I thought mRNA would be a limiting factor. But the one that is causing a bigger problem is the cholesterol. The FDA prefers that you use fully synthetic cholesterol, and it's the synthesis of cholesterol is not a simple matter. Of course, it's highly available in nature. We can get 100 milligrams or whatever out of an egg yolk, but the fully synthetic varieties are more difficult to obtain. So that has proved a bit of a bottleneck.
Okay, thanks, Pieter. So now I've got a few questions related to the importance of scientific platforms in the success in developing such a technology. So could you comment for you how important the support and the progress, I will say, made in Canada over the last 20 years in terms of financially supporting development of scientific platforms. So give us your perspective on this through the 35, 40 years of where you have been involved into this. You probably could identify a turning point during that time in terms of the importance of that support to develop a technology. And do you think we're in the exponential phase now with moving forward, or do we plateau? So what's your perspective on this?
Well, first of all, I have to thank CIHR, before that MRC, for, I guess it's been over 40 years of continuous support. So that's really played a big role. I guess the other part is that I realized early on that if you're going to get something into the clinic, then at some point, trying to do it through the university lab doesn't work. And I mean, the amounts of money are just too large. If you want to run a clinical trial, you know, it's millions of dollars. And so I've been able to start up companies to be able to raise substantial amounts of money to develop the, from say a bench scale where you're making, you know, lipid nanoparticles for one application or another, to the level at which, you know, will support a clinical program. And that, I think, is that transition that's really important, because you know, something that works at the bench level, there's very often big problems in getting it to actually work at a industrial level. And these are not minor, you know, people tend to think of scale up as being you know, straightforward, but it's not. You know, you have to have processes that are robust as you increase the scale of the operation. And we were lucky in a sense that we had the company involvements, which allowed us to really make that investment to say scale something up from making enough for a mouse model experiment to something that you could actually use in a clinical trial setting. So it's a very expensive process.
Yeah, no, certainly. So then a related question is, I guess, in terms of training of next generation of scientists in Canada. So again, your perspective on access to state-of-the-art scientific platform across Canada, across the different university, and those very important partnership with the industry. So are we going in the right direction? So give us your
Well, I've been encouraged on that a bit recently. I mean, I applied for a nanomedicines innovation network at NCE from the federal government, part of the NCE program, which was successful. And so we were funded in 2019 on that. And that's been a very good instrument, actually, for involving right across Canada. I think we have well over 40 investigators now. And so we're able to provide, you know, state of the art processes and you know, platform systems that people can then go and use in their particular animal model, or whatever expertise that they bring to the network. So that's that kind of support, I think, is essential. You know, where you're really enable that collaboration between groups. I mean, we're quite good at formulation. Other people are good at, say, a particular animal model, and ranging from, you know, some Alzheimer's model to a leukemia model, whatever it is. And then you can bring some, the combination, you can bring a reasonably sophisticated, you know, say nanoparticle solution, and test it out in a sophisticated animal model that is well characterized. And so that's the kind of thing that these centers of excellence can support, and it's that's an absolutely vital part of the process.
Okay, so my last question, Pieter, is it really about the lessons learned from the COVID-19 pandemic in terms of how important it is to support basic research and you know, expecting the unexpected. I guess that's a way to put it. So I guess again, based on your very broad experience, not only in the academic but the industry. So what are your wish moving forward in terms of addressing and navigating our way through the COVID-19 and the variants of concern, and with other pandemic coming along? What would you like to say in general in terms of supporting research in Canada moving forward?
Well, yeah, I think, you know, this is something that is, you know, very important, I think, to transmit that I don't think people quite realize that by supporting research in universities can have a dramatic impact on your personal health. And obviously the mRNA vaccine story is a very good example. And I think as scientists, we need to really get that word out there. I think some of the general population probably thinks, well, giving all these research grants to professors in universities, well, is that really something we're getting our money's worth for? And there's many ways that we can point out that that's true. But I think this is a particularly obvious one. The other part, of course, is that we were in the right place at the right time, but it was a very lucky circumstance in that sense. And when the results were announced last November, that was 95% protection against the infection by the SARS-CoV-2 virus. That was, to us, astonishing. It was it's very hard to predict something like that. And so it's a combination of good luck, but also the value of the basic science going on. That's in my lab's been going on for 40 years. It's proven to be pretty worthwhile. So.
Okay. Well, that's all I have for you, Pieter. Again, thank you so very much for your great talk and being such a fantastic ambassador of research for all of us in Canada. And I know you're going to be very busy with the next 5 years or so with all of the application of your technology platform. Thank you so much again.
Okay, thanks, François. It was a pleasure.
No problem. All right. So I hope you enjoy this amazing talk by Pieter, and also the very important insight into his perspective and related to research in Canada and the importance of scientific platform for Canadian scientists moving forward. Clearly, the breakthrough that he has done and his collaborator have done for the COVID-19 vaccine is one of the greatest example we can build on moving forward. This will conclude this session of the CNSP meeting, and then well, thank you again. Merci beaucoup d'avoir participé. And then we'll see you for the next session, which is the Quebec COVID-19 Biobank presentation, which should be starting at 1:55. Thank you all.
Hi, thank you for attending this Technobite. My name is Judith, and I will tell you about Mia Selavi's unique cycle for reproducibility. We offer consulting expert services in microscopy, imaging, and analysis for life sciences. We established in 2006 with the strong motivation that robust answers to scientific questions in medicine and research demand artifact-free, reproducible, and quantitative data. We have established a cycle for reproducibility, and this cycle begins with a number of databases. One database is for microscopy instruments themselves. We have over 500 instruments in this database, and these microscopes are described in their composition, their location, as well as the number of appointments for quality controls, preventive maintenance, repairs, etc. We also have a description, the contact information of the people involved in operating these instruments. Running quality control measurements on microscope involved a number of standard operating procedures that are now essentially described in a newly released confocal ISO norms. And we have a database for those procedures. And we also have a database for the specific test samples that are necessary to run the quality control measurements. This essentially describes the procedure as well as the reagents involved in preparing those samples. The next step is to go on the microscope and acquire data for one particular quality control measurement. Then the data is analyzed, again, in a very specific manner, and we have an output of this analysis in the next step of the cycle. All the data handling in these steps are done using the FAIR principles. That is, the data is findable, accessible, interoperable, and reusable. In order to get statistical significance, we need to acquire large data sets, and that involves automating computational processes. Therefore, we rely on open source tools to automate the process, but as well as having the possibility of looking at the codes if necessary. Therefore, the output is not only the result of the QC analysis, but we're also saving the computational processes that were used to generate this data. This brings us to yet another database, which is a repository of all the quality control data obtained in these procedures. And obviously this links back to the instrument. Therefore, when we repeat this cycle in time for one particular instrument, we now have the possibility of having a temporal measurements, looking at the performance of one single microscope in time. But the second advantage to this, because we have more than one instrument of the same type in the database, this procedure also allows us a system-to-system analysis of various quality control measurements. As a concrete example of where this procedure has led us in the past, I briefly want to mention about the reproducibility project in cancer biology, more specifically study number 20. And this study showed us that confocal microscopy reproducibility is challenging, but the good news is that there are tools available to help improve on that situation. So in conclusion, Mia Selavi has established a unique reproducibility cycle for microscopy equipment. This cycle allows two types of quality control analysis comparison. One of them is temporal, that is comparing one system with itself in time. And the other possibility is now to compare one system to another. Thank you for your attention, and I'll be happy to answer any questions.
So hello everyone, bonjour à tous. We'll start in a few seconds to make sure everybody is still here. Yes, everybody got connected, I see. So François, in the rush, forgot to say that the sponsor of our event make it possible to keep the CNSP membership fees very low. So there are also key stakeholders and partners with us that help support scientific platforms and platform scientists and administrators across Canada. So that was important to, that's why we have this TechnoBytes session to highlight some of our sponsors. So my name is Guillaume Lesage. I'm the treasurer of the CNSP and a co-organizer of this event. I'm also working at McGill as a grant officer in the Office of the Vice Principal, Research and Innovation. So it's my pleasure to be here today to moderate this session highlighting the success of the Quebec COVID-19 Biobank, la Biobank québécoise de la COVID-19, referred to as the BQC-19. So when we started to prepare this virtual event, it was obvious to me that the BQC19 should be presented as a success story. I mean, this scientific platform was created last year as a response to the pandemic and became operational very fast and despite very challenging times. As soon as the project was initiated in the spring 2021, several administrative units at McGill were engaged to coordinate and support the setup of the biobank. For my part, I've been involved in the participation and negotiation of the many agreements that regulate the activities of the biobank. And we'll describe this in a few minutes. But the presentation that will be given now will be given by three speakers. First, we'll hear Vincent Moser, who's the director of the Quebec COVID-19 Biobank. Vincent is a physician scientist. He holds the Canada Excellence Research Chair in Genomic Medicine at McGill. He came to McGill in 2019. Before moving to Canada, he held various academic positions in Switzerland, including chair of the laboratory department at the Lausanne University Hospital and vice dean for clinical research at the Faculty of Biology and Medicine at the Lausanne University. Vincent also spent 10 years running R&D in the industry at GlaxoSmithKline, so JSK, in Philadelphia, as vice president in charge of applied genomics and as the head of genetics for rare diseases. The second speaker is Pascale Léon. So Pascale is the manager of the Quebec COVID-19 Biobank. She received her PhD in life and health sciences from the Université de Paris 6 in France. After a postdoc in the US, she moved to Canada and worked for a few years as a scientist at the University of Toronto and then at the University Health Network. Since 2004, she has had several executive positions in the management of large research endeavors at the Ontario Institute for Cancer Research in Toronto, and then at the Canadian Consortium on Neurodegeneration in Aging at the Lady Davis Institute for Medical Research in Montreal. And then the last part of the presentation will be given by myself. Following the presentations, we will have a session of questions and answers as a panel. So we encourage you to ask your questions in the Q&A box at the bottom of the Zoom window, and we will answer them after the presentation. The title of the presentation is The Quebec COVID-19 Biobank as a Unique Research Platform to Study COVID-19. And to start the discussions, I would like to invite Vincent Moser.
Oui, bonjour à tous. Thank you very much, Guillaume and the organizers, for giving us, Pascale and myself, the chance to describe this platform in this very interesting meeting you have organized. So specifically here, we will focus on one platform, which is called the Quebec COVID-19 Biobank. The rationale for building up this biobank is shown on the next slide. On the left-hand side, you can see sort of the distribution of how people react when they have been exposed to SARS-CoV-2, the virus responsible for COVID. In green, you can see that the vast majority of people are essentially asymptomatic or have a mild disease. Some people in yellow have a symptomatic disease, fever, fatigue, loss of smell. Some people end up in the hospital with severe cases and breathing difficulties. And some of them, unfortunately, die, a fraction of them, probably 1 or 2%. And the question we're really asked to try to address is the following one. That's the same virus, SARS-CoV-2. So why do people react so differently to exposure to this virus? And this is not a trivial question. It's an important question because if we can better understand why some people react badly, for instance, we could imagine diagnostic tests to identify early who is at risk of developing severe diseases. And that can have immediate application. Imagine, for instance, a patient coming in the ER is coughing. If a test can tell, wow, be careful, this person will have a very severe outcome, then it's worth it to book a bed in the ICU for this person. if this person is deemed to have a favorable outcome, then it means that this person can go back home. So this is important in terms of diagnostic and of course in terms of treatment. As you know, there are limited treatment opportunities for this disease, and so new treatments are needed, even if, luckily enough, vaccines are now taking care of part of at least the problem. So the next slide shows you here the mandate we received from originally the provincial government here in Quebec. It was really the chief scientific officer and the head of Genome Quebec and the head of the Fonds de Recherche Québec en Santé who gave us this mandate, build up a platform, an infrastructure which will support research to identify the determinants of COVID-19 susceptibility. That means why do some people develop a disease? Severity, why do some people end up, for instance, in the ICU? And outcome, why do some people, for instance, have what we would call now long COVID? That means sequelae of the infection. In that sense, and that's where PHAC came in, the Public Health Agency of Canada, they wanted us to contribute to public health response to the COVID-19 pandemic. The next slide summarizes what we have done in a month. And we have put in place a steering committee of 10 people with various expertise, be it finance, genomics, ethics, legal, immunology, virology, and so forth. We designed the study, which I will describe to you later. We put a governance in place. We got part of the funding. The other part came later with PHAC. We wrote up the protocol and the SOPs. We got a fixed ethics approval to start the recruitment. And we also put in place the IT infrastructure to collect the data and have the samples properly identified. All that stuff was done within 10 days, actually. We received the mandate on 21st of March. Our first patient was recruited at the Jewish General Hospital here in Montreal 10 days later on April 1st. And that next slide, I think, by itself is a world record. Normally, outside COVID, you probably need a year to put that in place. Here, because of the vision, because of also the willingness of the stakeholders to get something done, but also, and Pascale will come back to that later, because of the existing platforms, it was really possible to do that. And that's really what I want to show in the next slide. All these things could happen because existing platforms. Now, I want to describe to you the study design. That's the next slide. Again, keeping in mind the mandate we received, that means finding clinical and molecular determinants of variable susceptibility, severity, and outcome of COVID, we designed this study as such. It's a prospective study with serial measurements over a 2-year period. It's an observational study where we collect extensive data plus blood at each visit. It's multi-centric across the province with 9 centers recruiting, affiliated with 5 major Quebec universities. So I think by itself, that's a tour de force to put all these people together. And here I express my gratitude to Guillaume and his team for making it feasible. It's broad. We are collecting individuals who are asymptomatic and patients who are severely ill. It's designed what we would call a case control. That means the capacity we have to compare people who are SARS-CoV-2 positive on PCR with people who are SARS-CoV-2 negative. And it's collaborative. We are not working in silo. We want here to be hooked with other initiatives like Concogen, CITF, CONCOV, and others, but also having the chance to collaborate internationally because the challenges we are facing are beyond what one single lab or province can really address. The next slide summarizes what we were able to do within a year. We have recruited 2,800 participants, now 3,500. That's almost 7,000 visits. For 2,000 of these visits, we have what we call a multi-omic profiling on the samples collected during this visit. That means a very detailed molecular assessment of the samples we have collected. This includes, for instance, sequencing of the host genome, measuring 5,000 proteins in each sample, measuring the anti-SARS-CoV-2 antibodies, measuring as well a variety of other parameters and small molecules. This is a platform again, and our goal is not to accumulate data. It is to share data after we have generated them rapidly and with high quality. So we have put in place some national and international collaborations, and so far we have contributed to 7 publications published or preprint. The next slide summarizes to you what are the numbers of patients we have recruited. On the left-hand side, you can see patients who are severely ill. In blue, people are cases, that means SARS-CoV-2 positive. In green, those who are controls, negative. And on the right-hand side, you can see the number of people who we have recruited with less severe disease or asymptomatic, be it PCR positive or controls PCR negative. So by itself already, we have one of the largest collection of patients properly recruited and consented for studying at molecular level the determinants of COVID. The next slide illustrates to you one piece of data. This is the results from what is called a genome-wide association study, or GWAS analysis. In this type of analysis, what we do is we interrogate variants spread along the human genome. Each of these variants here, and there are 2 million of them on this slide, are depicted by a dot. The location of the dot along the chromosomes, that means in the genome, is shown on the x-axis. For each of these 2 million dots or SNPs, variants, we ask the question, is this variant associated or not with COVID, severe COVID versus controls? At the bottom, you can see the number of people who had undergone these analyses, 12,000 people with severe COVID, a fraction of them coming from a biobank, the other one coming from a variety of laboratories around the world, and 1.7 million controls. And what you can see here on this called Manhattan plot is a series of towers. That means regions in the genome which are associated with COVID severity. Under the arrows, you can see on the left-hand side a peak in chromosome 3, and on chromosome 12, another peak with the label OAS1. I will give you now more information on the variant on chromosome 3, which is the next slide. At the bottom of the left-hand side, you can see the number of people who carry this very variant. You can see that 8,000 people do not carry the variant, 1,400 do carry the variant. So altogether, 14% or so of the population does carry this variant. On the x-axis, you can see the cumulative risk of ending up in ICU or dying from COVID, according in yellow to the presence of the variant. Yellow people do not carry the variant. Blue depicts the 14% of the population who carries this variant. And what you can see is that carriers of variant have a 40% increased risk of ending up in ICU or dying from COVID-19. So essentially, we have reached one of these goals. We have identified a genetic marker associated with outcome. On the right-hand side, you can see what is the risk associated with the presence of this variant and compare that with other comorbidities. Diabetes, DM, congestive heart failure, COPD, chronic kidney disease, cancer, obesity, and so forth. And what you can see is that the carriers of this variant have a risk, genetic risk, as much and as pronounced as the one that people who have diabetes or COPD or chronic kidney disease. So it's an important risk factor. And the question is now, shall we measure this variant in people so as to stratify their risk? As you remember, the next slide refers to the other tower on chromosome 12, the one associated with OAS1. OAS1 is a protein which is expressed by this gene. What Brent Richards at the Jewish General Hospital here in Montreal and a very large team of investigators is they have analyzed the function of OAS1, and in the next slide, they have demonstrated, the previous slide, that this protein does protect from COVID, and it may be then a new therapeutic target for COVID-19. So in summary, we have put together this platform. It's been very hard work. We are very glad we could have the chance to build it because of the vision and funding of our sponsors, thanks to the availability of existing platforms. And we look forward to continuing this big effort and making the data and samples available to the entire community. Thank you. Now, please, Pascale, the floor is yours.
Hi, everyone. Can you hear me? Okay. So in support of all these wonderful research results that, that Vincent just shown, there are a lot of, as you can imagine, a lot of work that's going on behind the scene. And I want to touch up briefly on two aspects: the governance of the BQC19 and all the platforms that are used to facilitate access to our samples and data, which are, as Vincent mentioned, really at the heart of our mission. So we do rely, I apologize for the typo, so the BQC19 relies on many platforms. These are just a few of the platforms, but there are very important ones. Of course, Nagano for everything that is ethics related, REDCap for everything that is related to databases. Our data are stored in platform under Calcul Quebec. The Biobank of the RSRQ in Quebec City also helps with, is used for the databases related to inventories of the samples. We have a collaboration with the National DLML in Winnipeg, the National Microbiology Laboratory, which is financed by the Government of Canada. And then the whole genome sequencing is done at the McGill Genome Center. And that's the place also where all the samples are centralized before being then dispatched to the different laboratories that will do are doing the what we call the core analysis, is all these different kinds of analysis on 2,000 samples that Vincent just mentioned. So in terms of governance, this is the organogram of how the BQC19 is organized. So the director, the steering committee is what Vincent mentioned was assembled in record speed, a group of about 10 people with different expertise. On the right-hand side, you can see the staff support. So it's a very lean organization with myself as the manager. We have an access officer who deals with, who oversees everything related to access to samples and data. And a very important role also is played by our coordinator, who is located in Quebec City and deals with everything related with the databases, which are really at the heart of the BQC19. Importantly, we have also in the first year assembled fairly quickly an international scientific council. And this is a group of international experts, very high caliber, that advise us on strategic direction for the BQC19. They meet every few months and give us very valuable insight of what's happening in the world, but also where we should go in terms of where we could go in terms of strategic plan. This is a group of experts in all sorts, again, of areas, from ethics to genomics to public health and epidemiology. At the bottom of the slides, you can see all these different areas of expertise. We call them dicastères in French, where that is represented at the steering committee level. And each member of the steering committee is responsible for one of these areas, one of these dicastères. So access data samples, the operation, the databases, etc. So the communication, psychosocial, pediatrics, ethics, legal, etc. So it's a very diverse group of experts and Vincent forgot to mention, I think, that we actually, when they started the BQC19 last year, they met every day, including Saturday and Sunday, for a few months. And then now, thankfully, thank God, we're down to one meeting a week, but we've been meeting every Thursday morning to organize things. So like Vincent mentioned, the core of the mandate that was given to us for the BQC19 is not simply, of course, to collect samples and store them, but to facilitate access to this BQC19 data and sample for the entire world, basically for the entire scientific community, not just in Quebec, not just in Canada, and not just academic, but also private companies. So the guiding principles around the access is to ensure that scientists have access to the biological material and data for the research efforts on COVID-19. And a very important point is that we being, let's say, Vincent or me or the steering committee, are not the one deciding on who has access to what. This is a very important aspect of the BQC19, but on the contrary, access to data and sample is under the responsibility of an independent access committee who is really solely responsible for granting access to data and sample. So we have a process in place which we facilitate, but which we do not tamper with or intervene in, and this all hinges around this independent access committee. So to date, we have, this is the situation in terms of data and sample access. We're really hoping to ramp up. We've been doing a lot of improvement to our processes around data and samples. And as we go along, as you know, with every platform, you learn from the previous cycle of access, and each cycle of access allows us to make significant improvement to our process. So in terms of data, we've granted access to 14 of our provincial collection, which represent the collection for all hospitals, and then 56 projects have been granted access to data in terms of the local collection that the hospitals have. And then in terms of samples, we're a little lower. We've had only 9 requests and 4 have been granted, but we are now embarking on a major communication strategy to really make the BQC19 better known and make the scientific community know that we have this wonderful resource that is in place. So this is just a snapshot of what has been happening, and I'll now turn it over to Guillaume for all the administration.
So thank you very much, Pascale. So Yes, when I invited Vincent and Pascale to give this talk, Vincent turned the invitation back to me saying, well, maybe you should present something about the administrator's role in setting up the BQC19. So that's how I came with this short presentation, the administrator's perspective. And in fact, as Vincent and Pascale showed, scientific platforms can usually be described at the scientific level with a great science and great questions to be solved and the great tools to solve those questions. So that's what Vincent presented. And they can also be described at the operation level. That's what Pascale did, showing the management team and the committees and the structure of the BQC. But well, Pascale, stole my punch, but she said that some things do happen behind the scenes. So that's exactly that was exactly my point. That at the administration level, some things do happen also behind the scene. So I mean here that the administration and the institutions do support the scientific platforms. And that's why I have put, as you can see at the bottom of the slide, the McGill logo close to the BQC19 logo, just to strengthen, highlight this support. In the McGill administration, 3 units are involved in this project. So there are some teams in the Office of the Vice Principal Research and Innovation, some people in the legal services, and teams in financial services. And these 3 units are involved in 5, what I would call 5 key administrative areas, which are a liaison with the funding agencies, a liaison with the partners institution, the financial matters, the material data transfer agreements, and the preparation of service agreements. And we'll go a bit more in more details in all of these, in each of these areas. On the next slide, please. So first, the liaison with the funding agencies. So our first role, the first role from the administration is to interface with the research team to validate the budget. As Vincent mentioned, the budget for the BQC19 has been set up in 2 stages. There was first a provincial involvement of $3 million budget, and then a second injection of money from the federal government for $7 million. So we had to build this budget by discussing a lot with the researchers, or a lot with Vincent and all the teams involved, to make sure that that budget was well set for 2 years to cover the salaries of the people, to cover the processes of sample collection, the way to transfer money to the collection sites, for the sequencing, for the additional analysis. So that budget was not an easy task to set. Then the administration has also been involved in preparing award agreements with the funding agencies. Again, the funding is coming from the Public Health Agency of Canada, the federal level, and there was a second agreement that was made with the provincial agencies, the Fonds de recherche du Québec en santé and Genome Quebec. And this is not a recurring agreement. Of course, these there's no recurring competition for that. It's one of its kind agreement. So the terms of the agreement had to be negotiated with the funding agencies, and that was some not tough, but some job and work from the administration. And then the administration, of course, is involved in ensuring and coordinating the reporting with the funding agencies, and make sure everybody is in touch with the PHAC and FRQS and Genome Quebec on time. Second, there's the liaison with the partner institutions. So first, at the onset of the biobank, we had to negotiate an interinstitutional agreement with many institutions. Again, there were in this agreement 13 institutions involving 45 parties. So this interinstitutional agreement is very important because it regulates and coordinates the collaboration between all these institutions, and it regulates the sharing of the material and data, and of course the sharing of the funds. So it was kind of, as Vincent said, a tour de force to have this interinstitutional agreement set during last year. Now we are in the process of amending this interinstitutional agreement because additional institutions want to join the BQC19. Administrative people are also involved in their participation to the governing committee described by Pascale, and more specifically, the Vice Principal Research and Innovation and the Deputy Vice Principal in Research and Innovation are involved sitting on this committee. And then lastly, the administrative people are involved also in verifying the ethics compliance and helping with the management frameworks. So the management framework is this document that that regulates all the activities of the BQC19, and it's part of the ethics package. And there's a team in the Office of the Vice Principal Research Innovation who is involved in reviewing the ethics document. And there's a lot of communication between the Research Ethics Board from McGill and the Research Ethics Board from the Centre Hospitalier de l'Université de Montréal, because most of the ethics documents are deposited at the CHUM. On the next slide, please. For the financial matters, of course, this area involves a lot of financial services. So people from finance were involved in opening the funds, so all the financial transactions. Before the award agreements were signed, McGill had to advance some funds to allow the project to start. So financial services were involved in this. And then once the award agreement have been signed and the money has been received, then they are of course managing the deposits. Financial people are also involved in overseeing the transaction and make sure that the transactions comply with the McGill policies and the funding agency policies. So they oversee all the purchases, the salaries, the payment to the sites, and the payment to the service providers. And lastly, finance people are involved also in the financial reporting. And I regrouped the two last points into a single area, a single administrative area, because these are all talking about writing agreements. So there's a material data transfer agreement and the service agreement. Our office and administrative people were involved in setting up the original material and data transfer agreement, so the template that is now used, as Pascale mentioned, by the access committee to grant access to the samples and the data hosted by the BQC. And as Vincent mentioned, there are some multi-omics analysis, and what Pascale called the core analysis. So these are downstream analysis on the samples from the BQC19. This can be immunological analysis, transcriptomics, sequencing, proteomics analysis. And this is done, this is regulated through service agreements that have to be written, and we're involved in this process too. And this is involving people at the CHUM, so academic laboratories, the IUCPQ, and facilities at the IUCPQ, which is l'Institut Universitaire de Cardiologie et de Pneumologie du Québec at Quebec City, and for example, the National Microbiology Lab in Winnipeg, which is a federal facility. So just to conclude that, as you see at the end of this short presentation, the institutional support can take many forms. Of course, we're not working full-time on this project, but there's a lot of support to help the project fly. And I highlighted a lot what McGill is doing, and of course, in other institutions, there are a lot of administrative people who are also involved in helping and supporting the BQC19. I would like to conclude by acknowledging all the people who worked on this project. So first, the BQC19 participants, which are the patients who gave samples, but also the control person who are not actual patients. Of course, we want also to thank the funding agencies, PHAC, FRQS, and Genome Quebec. we want to thank people from sitting on the steering committee and on the governing committees, because this is taking a lot of their time and they are directing this platform very well. And lastly, there's a lot of people who contributed to the success of this platform. So people who are nurses, people working on the collection side, people working to recruit the patients, researchers, technicians, administrative people who worked in the recruitment centers and in several universities. Then next slide, which is the end of the talk. And so I think that now I will thank you everyone for your attention. And Pascale and Vincent, I'm calling you back and we will go through a period of question and answers. So we'll have about 15 minutes of question and answers. So I'm seeing a first question from Marie-Ève Paquet. So it's a question for me, so I'm sorry. I'm starting the ball. So pour Guillaume, le lancement, le développement et l'expansion de cette plateforme a été extrêmement rapide. Mon expérience est que les administrations universitaires peuvent être plus difficiles à bouger rapidement. Est-ce que McGill a pu suivre le rythme si rapide? Est-ce que les nouveaux mécanismes ont été mis en place? So basically, she's asking that the setting up of this platform has been extremely fast, and she realized that usually universities are pretty slow in these processes, and she's asking how McGill was able to adapt to that rhythm, and did we set special mechanisms in place? So in fact, the process started in parallel. So the work already started while we were writing the agreements. So for example, the interinstitutional agreement has been signed in late October 2020. But the patients were already recruited and people started to work before that, well before that time. So the institutions did work as fast as they could, but there are so many people involved with legal services who really scrutinized the documents that we could not go faster.
Guillaume, can I make a comment on that question?
Mm-hmm.
I think the question is very good, and I think it illustrates what is needed to build up these type of things. We need to have a very important challenge to address. That was one. We had to get very strong support and hear from the province. The scientific en chef, Rémi Quirion, was behind us. He was saying, you've got my support, you've got money as well. Then we need to have a team dedicated. And here I was very lucky. With Carole Jabet, we assembled this team in 2 days. And like Pascale was saying, for 30 days, every day, we worked on trying to design the experiment and so forth. That was just very hard. But then we were very lucky that we had also universities, and the 5 universities here working together and essentially putting their effort towards this common goal. I think it's not pleasant to build these things. I think it's very tiring and we got exhausted, to be honest, but we had a big challenge to address. I think there is one lesson. This type of platform needs to be maintained, or at least, for instance, the interinstitutional agreements need to be kept somewhere so that if it happens again, and it will happen again, we are ready, much more ready than we were for this present pandemic.
Thank you very much, Vincent. I see another question coming from Sabine May. So she says, congratulations, what an achievement. And her question is, are you collecting samples from patients who contracted COVID-19 variants? And do you know if susceptibility depends on similar genetic loci as what you presented? So I guess it's probably a question for you, Vincent.
So thank you, Sabine. It's an important question here as well. We didn't know when we started this effort, we didn't know whether the host genetics, that means the variation in our own human genome, were associated with the severity or susceptibility or long COVID. We didn't know about long COVID. That was a bet. It was an expensive bet here. That means doing this analysis is expensive, it's complex, And essentially, I think what I have showed you is, for instance, yes, we did identify some variants, for instance, this chromosome 3 variant. Now, what we are looking for is, I think, along the question you're asking is, are there also some variants or some genetic makeup which explain, for instance, why do some people develop long COVID? We don't exactly know what long COVID means, but it seems to be that a fraction of patients who got COVID are have long-term consequences. For instance, heart problems, breathing problems, chronic fatigue, and you name it. We want to document this outcome, and we want to ask this question: can we predict them? If we can predict them, that means we can potentially intervene to help these people have these long-term consequences and get out of that. So it's not the end of the story. We will have breakthroughs in vaccines. We will have people who essentially will have these long-term consequences. Public Health Agency of Canada is also asking us, help us better understand these diseases, these outcomes, so that we can take at public health level the proper measures. That's another reason, in my opinion, to keep this type of platform. And I think that's also why I'm particularly grateful to have the chance to discuss this today as this scientific platform conference.
Thank you very much, Vincent. Now we have a question from Laurence Lejeune, which has been upvoted several times, so it's a burning question. Are there tools or processes developed that could be easily translated to other scientific platforms or equivalent collaborative projects? And if yes, could you identify them? Maybe that could be a question for Pascale.
So I think, like Vincent mentioned this, and I'm sure Guillaume will agree with me, the biggest achievement on my end, on my side of things, was to put together that interinstitutional agreement, because that meant a document that 11 originally, but institution agreed to sign. And of course, this had, in each institution, they had to go to lawyers, they had to go to administrators, etc. So it was really, you know, Vincent likes to call it the tour de force. I call it the nightmare, but you know, this no, but you know, this was a huge accomplishment, and that's the sort of, I think it paved the way to you know, when I started at BGC 19 in at the end of June last year, and I heard about this multi-party agreement, I kept saying to Vincent, this is not possible, like, stop doing that, you're not going to succeed. And he proved me wrong, and Guillaume proved me wrong, which I'm really happy about. And so this is, it's not really a tool, but I think this is the expertise that was put into the development of that document certainly could be shared with other platforms that are facing the development of this really, really complex agreement. And maybe it's just a question of, you know, sharing our experience and how little by little we manage to solve problems and talk to institutions and solve these all the issues that were millions of them. Another interesting aspect that I mentioned during my presentation with this independent access committee, this is also something, we're not the only one in the world to have this kind of access committee, but also this is also all our procedures around accessing samples and data certainly could be shared with other platforms, because a lot of thoughts went into them, and they've improved really greatly in the past 6 months, and they continue to be improved. So that's the sort of experience and expertise and tools that we could share.
Guillaume, can I make a comment?
Of course.
Just to follow on what was said. We are discussing about platforms here, and everybody probably around the table acknowledges that platforms are expensive and sustainability is always an issue. I would, if in the audience you have people whom you know who may be interested in accessing our data and samples, please let them know that we now have, Canada and Quebec have paid for this resource. It's available for researchers, and our success And the expectations from our patients, really, and from public health and the taxpayers, is that we can bring some answers to these terrible challenges we have been facing. So if you know of any scientists or people interested in what we have done, or if, like you were saying, the SOPs, all the infrastructure we have put in place can be of any help for other initiatives, visit our website. All the documentation is there, including how to access our samples and data.
Okay, well, thank you very much for answering these questions. I think that I don't see any additional questions in the question and answers. I think we might be done with this session because we need to then wrap up. So Vincent and Pascale, I thank you very much for your time in preparing this talk and for your great talk and great work on this project. I mean, I'm working with you. It's a pleasure to work with you. So this concludes the first part of our meeting. Now we will take a small break. So we encourage you all to get up, stretch, refill your beverage, and then to return at 3 p.m. After the break, we are reconnecting on Zoom meetings. So to find the link, you have to go to the schedule tab in the guest board, and we will begin again promptly at 3 p.m. So see you soon.